So today we're going to do energy, enzymes, and transport. All of these ones are basically interconnected. Energy. What is that? Energy is basically defined as capacity to do work. That's how energy is, basic definition of energy. Now, energy comes in different forms. In the cell, the only form of energy that the cell can use, it's called ATP. Cars use what form of energy? Gasoline. For your stove to heat up your food, what form of energy do you use? You use the gas methane, which will burn and releases energy. Now, cells work exactly like small factory, Why? Because there's a lot of reactions taking place in there, and they have stuff that needs to be produced to be sent outside the cell. And sometimes, or not sometimes, and actually sometimes, cells require stuff that's needed inside the cell. In this case right here, we'll see glucose. Glucose comes in from where? From the blood vessels, from our food actually, then it ends up in the blood vessels, and then glucose it's taken up in the cells where it's going to be broken down to release these units of energy. Now, glucose is the main source of energy inside the cell. Anytime the cell needs energy, looks for glucose, but also it can look for proteins and fats. Now, the result of all these reactions that take place inside the cell is to maintain the cell at equilibrium. This process, it's called homeostasis. So when you keep it at equilibrium, the process is called homeostasis. Now, energy, two kinds, potential, kinetic. Now remember, there is many different forms of energy. Okay, out there, chemically speaking, but there is two main kinds, potential and kinetic. Kinetic stands for movement. Kinetic, it's movement. Potential, it's static. This is movement. So potential energy, it's stored energy or static energy. Kinetic energy is the energy of motion. Now, an example right here, you see both forms of energy. The water behind the dam right here, it's stationary. Now, this water here stores a lot of energy. It stores a lot of energy. Now, this energy, it's called potential. So the energy behind the dam, it's called potential energy. As the water escapes the dam right here at the top, and it moves down these pipes, and turns these turbines right here to produce electricity, the energy that moves down here, so from stationary water, it turns into movement, which is kinetic energy. So you go from stationary or potential to kinetic energy. Here's an example. A dude, I call him always usually Lance Armstrong, bicycle up the hill. Now, as he's at the top of the hill right there, he stores a lot of potential energy, a lot of potential energy. I always joke this is where he's contemplating what's he gonna do after he got busted with that he was using steroids during the, or like enhancement during the Tour de France. But here it's potential. Here is kinetic. So remember, kinetic has to do with movement. Now, how do we measure the energy? For all units of measurement, there is some type of unit that it's used. The most used one for form of the most used unit for the form of energy, it's called joules. Now, if you talk about food, we don't use joules. There is countries in this world where they use joules, but in America, you will use calories. We use calories. Now, what is calorie? This is the small calorie, by the way. This is the small calorie. So a calorie is the amount of heat required to raise the temperature of 1 gram of water by 1 degree Celsius. So basically, I give you a gram of water in a small tube. The energy that it takes to raise the temperature of that water by 1 degree Celsius, it's called calorie. Now, actually on foods, what we see, it's the kilocalorie. Kilo means 1,000. So 1,000 small calories equal 1 large calorie. So there is the small calorie, and then there is the large calorie. This one, it's also the kilocalorie. Okay, doc? So this is the kilocalorie or the large calorie, and this is the small calorie. Two laws are used for energy or in physics. Energy transforms always from one form to another. So the first law states that the energy in the universe is constant. Basically, nothing can be neither disappearing, neither showing up from nothing. You cannot create energy, cannot be created or destroyed. Energy, all what it does, it changes from one form to another form. For example, the energy in gasoline, it changes from gas into mechanical energy, which actually helps move the wheels of the car or help move the car. Second law of thermodynamics, it states that energy cannot be changed from one form to another without losing some usable energy. So basically, this refers to efficiency. When energy goes from one form to another form, it's not going to be 100% extracted, but you're actually going to lose some. How do we lose some? It's mainly in the form of heat. For example, same example as earlier, when gasoline turns from gas to actual kinetic energy or mechanical energy to spin the wheels, a lot of that energy in the gasoline, it's wasted in the form of heat. That's why your engine gets very hot. That's why the engine of the car gets very hot, because as the energy from the gasoline, it's broken down to be turned or transformed into mechanical energy, a lot of it is lost in the form of heat. So the most efficient car engines these days, they use only about 40%, maybe a little bit more, just 42% of the energy they can extract. Those are the most efficient. Back in the days, the engines could extract between 25 to 30% of the energy stored in gasoline. Now, as the energy is lost in the form of heat or any other form, basically through conversion, you increase the disorder. This is called, or the measurement of disorder, it's called entropy. Entropy. For example, you burn wood. Wood, it's very nice arranged before you fire it up. Once you heat it up, a lot of that energy is released in the form of heat. A lot of that energy is released in the form of heat. Now that heat is going to spread everywhere. It's not going to be organized as initially the energy was organized or the molecules were organized in the wood. So when you go from one form to another, you actually lose some of the energy, but at the same time, you increase the disorder. That measurement, it's called entropy. Now, when you look in the next picture, you can see 2 bedrooms. Now, which one has more entropy and which one has less entropy? Now, this one to the left right here has less entropy. Why? Because it's just very organized. This one that has more disorder, it has way more entropy. Now, how is that how is that making sense, basically? Because the higher the entropy, the more stable it becomes. If I walk in your bedroom right now, I bet you it looks like this one right here, not like this one to the left right here. It takes way more energy to actually keep it neat and clean versus just having it all messy. Now, the reactions that take place when the actual energy is formed from one form to another, they are of 2 kinds: exergonic The types of reactions are exergonic and endergonic. Now, exer means outside, in exterior, outside. So basically what it means here, in an exergonic reaction, energy is released from the type of form of, or the chemical itself. So it's released. So it's a chemical reaction where the reactants release energy. Now, we're going to learn today what goes into a reaction. If you have a chemical reaction, for example, A plus B, you get C. What goes in, they are called reactants. What comes out, it's called the products. So while these two react here for an exergonic reaction, products have higher energy. Products have higher energy. Products, products have less energy, not higher energy. Products have less energy for an exergonic because these reactants here have high energy. So basically, while they react, products lose energy. So products are low, reactants are high in energy. An example would be cellular respiration. As you start with glucose, right? Glucose contains all the energy that it needs. While you break down glucose, it turns into carbon dioxide and water. These two products at the end have very little energy. So the reactants had high energy, products had low energy. When you talk about endergonic reaction, basically it goes inside, you input. So here it's the opposite. A plus B turns into C. Here you input energy. When you input energy, basically the products have high energy because they picked it up. So the reactants have low energy and the products have high energy. So this is called endergonic. Now, an example of this reaction right here would be photosynthesis. Basically, for photosynthesis, you have the plant right here. Plant picks up the energy from the sun right here. Plant picks it up and it turns it into glucose. So this is basically an example of endergonic reaction. Another endergonic reaction. If you heat up your milk at the beginning and milk at the end, after you heat it up, which one has more energy, basically? Definitely the one at the end, because you input energy to warm it up. Now, all this one is needed, whatever we discuss, or it's used because of metabolism, takes place because of the metabolism. What is metabolism? It's basically the sum, or basically all the biochemical reactions in the body. All the biochemical reactions in the body or the cells are part of metabolism. All of you probably talked about metabolism or heard about metabolism. You say you have high metabolism. High metabolism means what? It means that you break down chemicals or stuff that you eat much faster. Now, as I mentioned earlier, what goes in the reactions are called reactants. What comes out are the products. Also, I mentioned that ATP is the main energy or the currency of cells. The main is the energy currency of the cells. Cells cannot use anything else except ATP when they need energy. Now, how is ATP formed? The arrow right here is misplaced. But ATP turns into ADP and phosphate when it's broken down. When you put it back, ADP plus phosphate turn into ATP. So here, to go forward, this reaction is exergonic. This reaction, when you go backwards, when you get these two reactants to turn into ATP, which has a lot of energy, this reaction is endergonic because we input energy. Now, ATP, or the structure of ATP. ATP is similar to a nucleotide. Now remember, nucleotides make up what? The DNA and the RNA. Now, there were 4 nucleotides, ATCG for DNA, AUCG for RNA. Those are basically stable. They cannot be broken down. ATP, it's renewable or recyclable. If you look right here, ATP turns into ADP plus phosphate when energy is needed. When you have plenty of energy or you extract the energy from glucose, ADP plus phosphate plus about this much calories per mole extracted from glucose form or turn into ATP. Now, ATP, it's made up of sugar, phosphate, and nitrogenous base, exactly like the nucleotide. Now, the interesting part is there's a little bit difference right here. That nitrogenous base right here, it's the adenine. So basically, this ATP molecule, it has the 3 phosphates versus 1 phosphate that's found in the nucleotide. It has 3 phosphates. It has the same sugar that's found in the RNA, and it's interesting, as I mentioned, that it contains a nitrogenous base, and it's only adenine, not the other ones. Is that clear? Now, there's other different forms of energy, CTP, which could be cytosine sometimes, or it could be GTP, which is guanosine or guanine. Now, the main difference between nucleotide and the nucleic acid or the nucleotide, it has 3 phosphate groups. Now, all the energy that we talk about, it's stored in this bond right here. This bond, when the cell needs energy, it's gonna be broken down through the process of hydrolysis. And we learned this one a while back. The process of hydrolysis. When you put this bond the way you see it right now, when we put it back, the process is called dehydration. So this bond, when it's broken down right there, you go from ADP plus a phosphate, this is the phosphate, and turns into ATP. ATP, when energy is needed for the cell, it's gonna be broken down to ADP plus phosphate. So you see right here, this reaction that takes place right there, it's called hydrolysis. Now, in our body, we don't store ATP. ATP is stored in very small amounts. And it was found recently, actually, that we store some ATP in the muscles and only in the muscles. Why? Because muscles need immediate form of energy. Muscles need immediate form of energy. So they store for like maybe 1 to 2 seconds, enough ATP so it can start extracting from pyruvate or glucose. We're gonna learn later about this step. We're gonna actually next time. Now, the more stable form of energy stored in the body are carbohydrates and fats. Carbohydrates we store in the liver, we store some kidneys and muscles. Fats, where do we store them? Everywhere throughout the body. So remember, carbohydrates and fats are more stable, so it's long-term energy. ATP is very unstable, so it's very short-term for just a few seconds and only in the muscles. Here, you can see the cycle or the recycling of ATP. ADP plus phosphate turns into ATP when energy comes from glucose. During cellular respiration, the process is called. Now, when energy is needed for the cell, boom, energy is released. Here, energy is released. And it turns into ADP and phosphate. And this one is going to keep going back and forth. So basically, ATP, it's recyclable. Now, production of ATP has several benefits for the cell. Which one is that, or what are those? Basically, ATP, it's used for all the reactions inside the cell. When the ATP is going to be split, there is only enough energy, the amount of energy released, or there is enough energy for each of the reaction. So if I have reaction A plus B and it needs energy to turn into C, I add 1 molecule of ATP. Most of the reaction in the body require about 7.5 kilocalories. So if you look right here, this is exactly how much basically it's stored in the molecule of ATP, in the bond right here. In the bond right here, there is basically about 7.5 kilocalorie. And that's why basically through evolution, ATP came along. Because rather than start breaking down large molecules which contain or store large amounts of energy, molecules that store large amounts of energy, I'm just going to have a lot of waste. But since the reaction required very small amounts, about 7.5 kilocalorie, ATP came along, or ATP developed. And then all the reactions in the body, they require energy, they input ATP. Now, if you need more than 7.5, you bring a second ATP there, and then you release another 7.5 kilocalorie. If you would bring a molecule of glucose, glucose stores a lot of energy, stores about 1,000 kilocalories. Now, if I bring a molecule of glucose here and I try to break it, I waste a lot of it since this reaction requires very small amount. So if this reaction required only about 8 kilocalorie, ATP is way more than enough, 1 ATP. If I would bring glucose and I start breaking it, I actually waste a lot of the energy. So that's why ATP, or the body evolved, or this new form of energy formed. Chloroplast use the solar energy to convert water and carbon dioxide into carbohydrates, or you call them sugars. Now, in our body or in the plant cells, cellular respiration is the process in which this other organelle, mitochondria, breaks down these carbohydrates here, this one from here, to produce ATP. Now, this process right here, it's cellular respiration. The process right here at the top, it's called photosynthesis. How do humans contribute to the flow energy? Pretty simple. We use what? Glucose, which it comes from plants, and we use oxygen, which also is produced by plants. And we produce carbon dioxide and water, plus the energy that the body needs to grow and develop and for homeostasis. When plants need food, they're not going to go and look for salami or In-N-Out or any other type of food. They just go straight up to the air and they pick up carbon dioxide and they take water from the soil and they turn it back into glucose and oxygen with the help of sun, because they are capable basically of turning carbon dioxide and water. Plants are able to turn carbon dioxide and water into glucose. Where? In the chloroplast. Now, oops, right here. Metabolic enzymes. What are enzymes? Enzymes, by definition, or most of the reactions that we talked about until now, they are catalyzed by enzyme. What does catalyze mean? Catalyze means basically that it helps them, speed up the reaction. Without enzymes, the reactions cannot take place. Without enzyme, what's going to happen? Everything will die, because basically we cannot extract the energy, or we cannot assemble stuff in the cell. Remember, in the cell you need both ways, break down stuff or assemble stuff. Now, enzymes are molecules that basically function as catalysts by speeding up the chemical reaction. How much? Actually, it's up to 10 to the 9 time here, to 1 billion time actually. Now, for enzyme to stay active and to work, they require 2 important things: temperature and pH. It's very, very important for the enzymes. If the temperature and pH keep changing, then the enzymes will be denatured. Denatured, it means that an enzyme that takes a certain shape, like for example, right now, takes a certain shape, it will take a different shape. To give you an example of denaturation, an egg, before you boil it, the egg white, it's very viscous. It's like watery, jelly. Once you heat it up, you did not destroy the egg. All what you did, you just heat it up. Now the egg white looks white now. Why is that? Because the protein that makes up the egg white part or the white part, it's going to be denatured. So the process of denaturation, it's when an enzyme or a protein changes shape or conformation. And we talked in the lab about that one. Now, temperature for enzyme, for human beings at least, the enzyme requires a temperature around 37 degrees Celsius. That's why our body temperature is maintained constant at 37 degrees Celsius. That's how doctors actually know when we have some type of infection in the body, because our body temperature rises. When you have a bacterial or a viral infection, basically your body tries to fight the infection. How do they fight the infection? Raise the temperature. By raising the temperature, they start denaturing the proteins, the important enzymes that make up the bacteria or are required for bacteria or virus to properly survive and to properly reproduce. Now, if you raise the temperature, the enzymes that make up the bacteria and the virus, or the enzymes that are needed for bacteria and virus to survive, will be actually denatured and then basically will take over. Is that clear? Your body will take over. Now, enzymes also require the pH around. Now, probably, so whoever wants to be a nurse, or probably you guys heard until now, you guys heard the physiological pH. What is that? That's basically the pH that the body maintains it inside. Inside our body, we maintain a constant pH. That physiological pH in the body, it's about 7.3. So now I want you to tell me if you believe that this 7.3, it's basic or acidic. So remember this one, it's around 7.3, the physiological pH, and you have to determine if it's basic, slightly basic, or it's acidic or slightly acidic. Now, in the body, all these reactions go through metabolic pathway, It's not just one reaction. Most of the time, you start with one reactant, for example, sugar. Sugar is going to be broken down through many reactions during the process of cellular respiration to turn into the final product, which is carbon dioxide and water. Now, why do we have to go, or why does the cell go through many reactions rather than one? For one main reason. As you go through small reactions, or to many reactions, not small reaction, through many reactions, you slowly release the energy without losing too much, Basically, by having pathways or many reactions here, starting with the reactant at the beginning, you extract more of the energy. If you would just have one reaction, you would waste a lot of the energy. You would waste a lot, or the body will, or the cell will waste a lot of the energy. Now, how do enzymes work? Enzymes look something like this. This is a form that you see most of the time, the enzyme. So it's the enzyme itself, which has basically a site right there, which is called active site. Now, what is that site? As the name tells you, it's active. That's where the substrate or the reactants, I forgot what I seen somewhere here. The substrate or the reactants basically come and fit in here. So if you have A turning into B, this is the final product. This A come and fits right in there in the active site. Then the enzyme does the trick or its job and turns it from A to B. How that process takes place? Basically, by lowering the activation energy or energy of activation. Now, what is that energy of activation? For reactants, which are right here at this level, you see right here the energy level, to turn into products, which are lower in this case right here, they have to go over a hill. Now that hill is pretty high. It's kind of like you guys want to reach 10 freeway from Monsac. You take Grand, but before you get to the freeway, you have to go up that hill. And it's very hard to go up that hill. That's in terms of enzyme, it's called energy of activation. To actually reach the top of the hill after which the reaction takes place, it needs to input or to use some of that energy. So that's called energy of activation. This is for a reaction that is not catalyzed by enzymes. A reaction that is catalyzed by enzyme, look at the hill versus initial one. So basically what enzymes do, they lower the energy of activation from here to here, from very high to very low. Now, are enzymes used in the process while the reactants are turned into products? No, enzymes are not used during the process. So after the enzyme does its job, eliminates or kicks out the product and picks up another substrate. So this enzyme, basically, you put the substrate, turns into product. Substrate. So substrate goes in there, turns into product. Now, once this product forms, another substrate comes in and another substrate. And this enzyme is going to stick around as long as it's needed. So sticks around as long as it's needed. It's just not going to go away. It's just going to stick around. When it's not needed anymore, when it reaches the end where, okay, cool, we have enough product, then the enzyme will be denatured and degraded. And then it's going to be taken out of the cell. You could see right here how the enzymes work. Enzyme, active site, in this case, sucrose, comes in here. You see sucrose is made up of two of these molecules, fructose and glucose. It comes in here, fits in the active site. Through the process of hydrolysis, breaks down this bond right there. Now you got these two products, which is glucose and sucrose right here. Sucrose and fructose, not sucrose, fructose. Now, then they are kicked out, fructose, glucose, and the enzymes goes back and picks up a new substrate, and that's what is going to keep going and going as long as there is enough substrate available or you have enough product. Now, how do enzymes work? Enzymes have, not how they work, sorry, the models of the enzymes. How do enzymes actually work, kind of. But the model of the enzyme could be of two kinds, lock and key and induced fit. Lock and key, as we should know by now, it means that there is one key for one lock. So the same thing right here. There is one key, one lock, one substrate, one enzyme. So these two only together they work. There is no other substrate that can fit in here. There is nothing. There is nothing. If the substrate looks something like this, it's not going to fit in there. But this only this substrate right here. That's why it's called lock and key. There's only one enzyme, one substrate. So it fits perfectly. Induced fit, as the name tells you, induced fit, basically you induce it, you push it to fit. You push it to fit. So in this case right here, if you look, these substrates come and try to fit in here, but the active site doesn't look like this one right here, that it fits perfect with the substrate. So what is it gonna do? These two substrates come in here, they push so they can fit in there perfectly. Once they fit, they can turn into product. In this case right here, the enzymes or the active site of the enzyme adjust its shape. So basically you can have many substrates that fit in the active site. So it's many substrates that fit in the active site. Many substrates that fit in the active site. Now, these enzymes actually could be also inhibited. When they are not needed anymore, they will be inhibited. Or sometimes we use medications, or we use other enzymes which will compete with the enzymes in our body or block them from actually working. Medication, sometimes like headache medication, when you guys get the headache, basically there is some reactions that takes place in the brain which produce the wrong product, which will give you the headache. Now, if I will block the enzyme that it's involved in production of the wrong product, I can block the actual headache, me getting the headache. So that process is called enzyme inhibition. So what happens during enzyme inhibition? Basically, you block the substrate to bind to the active site by using an inhibitor. So you block the substrate, this is your enzyme. You block the substrate that comes to fit in here, you block it with an inhibitor or something that might look like the actual enzyme itself. Looks like the substrate itself. So basically blocks the substrate from binding to an enzyme with another inhibitor. Now, there is two types of inhibition that I will teach you, competitive and non-competitive. Competitive inhibition, it means that it competes with active site. Competitive competes with active site. Competes active site. Non-competitive inhibitor, it basically binds to another region on the enzyme and it changes the active site. Changes active site. So basically, competitive sticks directly into the active site itself, and now it blocks the substrate to come and fit here. And non-competitive, it binds to another side of the enzyme. By pushing this side right here, it changes the active site. By changing the active site, this substrate cannot fit anymore in the active site. Now, cell transport, it's a form of transport or how stuff moves inside and outside the cell. Now, who regulates this cell transport or this movement? It's the actual plasma membrane. Now, we learned last time that plasma membrane was a lipid bilayer which had proteins embedded and also carbohydrates. So by having those proteins and those carbohydrates and the fact that it's a lipid bilayer, the plasma membrane becomes basically semi-permeable. It will not allow everything to move, but it controls what moves in and what comes out. Now, by that, it means that the membrane is differentially permeable or semi-permeable. Permeable, basically substances move freely, but only certain of them move in and out. They are controlled which substances move in and outside the cell. Now, they can enter the cells or exit the cell 3 different ways: passive, active, bulk. So all of you heard or know what bulk means. It means whole or large, like large amounts. Like when you go to Costco, you're not going to buy just 1 pack of gum. Like when you go to 7-Eleven, you buy a whole packet. You buy it in bulk. You buy a whole huge packet with about 30 or 40 or 50. How many packets are in there? So the same thing. Cell transport could be in bulk. Now, we've seen this one before when we talked about vesicles or the vacuoles. Vesicles or vacuoles which will stick or which will fuse with the cell membrane or the plasma membrane, and then they will take in or eliminate the stuff outside the cell. Passive transport. It's the one that does not use energy. For example, the real world. I'm going to put a lot of cologne in me, and then outside the classroom, and then I'm going to walk inside the classroom. Guess what's going to happen? What is the first thing that you guys are going to notice? Or you're going to smell? You're going to smell the cologne, start smelling the cologne on me. How? People close to me immediately will start noticing. People further away, it will take a little bit, but it will get there. So basically, for passive, I did not do anything. All what I did, I just put the cologne in me and I walked in. Now these molecules, due to Brownian movement, they just gonna move away. The molecules of Cologne, they move away from me, and they spread everywhere throughout the class because they will bounce or they will collide with the air molecules, So this is a type of passive transport. Active transport is basically when I use energy to move stuff around. It's when I use energy to move stuff around. Now, a small or a quick example of passive transport is diffusion. Diffusion, basically what it does, it just moves chemicals from high concentration, certain chemical, to low. So it goes from high to low because the membrane allows it. Water, it's one case of passive transport. Water can easily move in and out the cell, in and outside the cell. Facilitated diffusion. It's basically when the molecules cannot pass through the membrane, but rather they cross through a protein right there. Exactly the same way as diffusion. They move from high concentration to low concentration. How long they gonna move? As long as the concentration is not equal between inside and outside. As long as the concentration inside and outside is not the same, molecules will move always from high concentration to low concentration, always from high concentration to low concentration. So simple diffusion, it's basically when the solute, which is the substance dissolved in the solvent, move from an area of high concentration to a low concentration. Basically does not require energy. Now, what's very interesting, a lot of the molecules that are non-polar can easily move through the membrane. Why? Because this membrane is semi-permeable and it's lipid bilayer. So it's a lipid. As we guys learned before, lipids are non-polar molecules. So non-polar mixes with another non-polar molecule. So examples would be oxygen, carbon dioxide, and water can very easily cross the membrane from high concentration to low concentration. You can see here the passive transport or diffusion. Osmosis. Osmosis, it's basically, and we're gonna do it this Friday in the lab. Osmosis, it's just movement of water across a semi-permeable membrane. So basically, I'll give you a tube. You put in the tube molasses. You're gonna put 80% sugar in the tube. It's molasses or sugar. Then we put this membrane right here. We attach it right here. We put this membrane, and then we put this one in water. We're gonna put it in water. So you have 80% sugar in here, and you put it in water. What's gonna happen? Basically, water moves from outside inside here, and then the volume will rise. Why is that? Because if it's 80% sugar, only 20% is water here. Outside here, outside the tube right here that we put, water, it's 100%. So remember, water freely moves or easily move across the membrane from high concentration to low concentration. So this is called osmosis. Now, let me give you examples of real things. Actually, let me do it over this one, and then I'm gonna give you an example of real things. The effect of osmosis on cell. Now, we use specific names when we look at osmosis and the cells. The process or the term, it's used tonicity. It's basically tonicity. It's the ability of a solution to cause a cell to gain or lose water. So basically tonicity, tonic, it refers ability to gain or lose water. Now, when we have an isotonic solution, concentration outside and inside, it's the same. This is isotonic, same. Let me write right here. Oops. When we have hypertonic solution, it basically tells us that the solute is higher outside. So right outside the cell, outside the cell, the solute is higher. So now what is going to happen? It means that I have a lot. Example would be, I just ate something very salty. All of a sudden, I become thirsty half an hour later. Why is that? Because a lot of that salt end up in my blood. Cells which are made up of a lot of water start releasing water outside. Why? Because they try to equilibrate the concentration of water inside the cell and outside the cell. Because right here I have way more salt than inside the cell. So now water is going to come out. Once the cells start losing their shape and they start shriveling up, in this process here, shriveling, or if it's plant, it's called plasmolysis, what's going to happen? That's when they start sending the message to the brain, hey, this person needs water because the cells are becoming shriveled and I don't want that shape. If the solution is hypotonic, indicates basically a higher concentration of solute inside, So right now my concentration is way, way higher inside the cell, right there. Now, what's going to happen here? What do you think, guys, is going to happen? Water actually starts, boom, moving inside here, and water keeps moving inside. Guess what's going to happen? Your cells start growing and growing and growing and expanding, and sometimes they actually could plasmolyze or die or basically lyse or basically explode. Now, a few years back, there was a competition. It was like about 9-10 years when first time the Wii came out at a radio station in San Francisco, and they made him sign a release of liability. It was a competition. Who drinks the most water in the shortest amount of time, which is I think 10 or 15 minutes, wins a Wii. Now, one of the persons in the competition died. Why did they die? Because they drink too much water. Water was in high concentration outside the cell. Water started moving inside the cells. Now, once they start moving inside the cell, a lot of those cells start actually exploding or dying. Now, what's going to happen? If important cells in the body, like liver cells, kidney cells, will start dying, then you're going to end up with big problems, big troubles. So that person, I think it had kidney failure because in that short amount of time, kidneys could not eliminate the water. In 10 minutes, if you drink 2 gallons or 1.5 gallons of water, you will most likely have a high chance of dying because it takes a longer time for actual kidneys or the body to expel the water, the extra water that we drink. Now, if you drink too much water, remember, your cells are hypertonic. So then the solution is going to move, the water it's going to move inside, and then the cells will blow up. When you drink salt, it's the opposite. Now concentration is out, your cells will not explode, but you will definitely have to drink water because you need to eliminate that salt. Now, that's why you cannot drink salt water, or when you go to the beach, you can drink ocean water because ocean water contains a lot of salt, which will lead basically to thirst, and then your cells will shrivel up, and then if you don't have enough water with you, you will die eventually. Active transport is the last type of transport and infers that it requires energy. Now, what form of energy there is inside the cell? There is only one form, which is called ATP. So for active transport, you need, or the cells need, ATP. Now, active transport also requires a membrane protein. Why? Because you need to pass it through the membrane. Remember, the membrane is lipid bilayer. Remember, phosphate with 2 heads. So you cannot push it through the membrane. So then you require what? Somewhere here to get some type of protein, or here, some type of protein. Now this protein, what it does, will allow the molecules to move. Now, if you look right here, this is the protein. That's what it looks like. Now, for this protein to be able, or for this chemical to be able to get inside, I don't know what which way it goes in here, but inside or outside the cell, what it does, this part right here, it's closed. Now, what you have to do, you have to clip these ones. Basically, you have to put the solute inside, or the chemical inside, in this case right here. Then you bring the ATP, which will push these ends right here next to each other, close them. So then the solute in here, or the chemical in here, it's going to be expelled. Once the chemical is expelled outside, this ending right here comes back to the initial state. Now, there is no more energy right there, because remember, to keep this one, or to attach these two ends right here, you need energy. That energy comes from ATP. Once there's no more energy, these ones come back at the end as the initial shape. Now, here, I want you guys to go over these sample questions, which kind of look like the exam ones. And I think there's 3 of them that I put here. Yes, exactly 3 of them. Answer them, and if you have any questions, you can ask me on Friday about them. Enjoy the reading, guys, and that's the lecture for today.